Understanding the pathology of Alzheimer's disease. Alzheimer's disease is a form of dementia characterized by gradual, progressive impairments in memory, reasoning, perception, and communication. Neuropsychiatric symptoms such as depression and apathy are core features of the disease. Agitation and aggression also often develop, which is challenging for patients and caregivers. The disease culminates in significant loss of independent function and eventually death. One study has shown that 75% of patients diagnosed with Alzheimer's disease die within 10 years of disease onset. Globally, around 50 million people suffer from dementia. Alzheimer's disease may account for up to 70% of this number. It is considered the most common neurodegenerative disease of aging. Age of onset ranges from 40 to 90 years of age, with a steep increase in prevalence with increased age. Onset of Alzheimer's disease is affected by multiple risk factors, with increasing age being the biggest risk factor. Genetic factors, age-related events, and pathological conditions may all affect risk of onset. The role of amyloid and tau in Alzheimer's disease. The two core pathological hallmarks of Alzheimer's disease are amyloid plaques, formed by extracellular collection of amyloid beta peptide, and neurofibrillary tangles, which are triggered by intracellular assemblies of hyperphosphorylated tau, a protein normally involved in microtubule creation. The amyloid cascade hypothesis suggests that toxic levels of amyloid beta trigger changes in tau and consequent tangle formation. Seeding and spreading of tau tangles may drive Alzheimer's pathology. Microglia take up and break down tau released from neurons, but high tau levels may disrupt this process and thus drive neurodegeneration. Preclinical data indicate that amyloid plaques and tau tangles aggregate and together impair the function of neural circuits. Neuronal loss is also seen, possibly a result from oxidative stress, microglial activation, and apoptosis, in response to amyloid plaques and buildup of tau tangles. The staging model of Alzheimer's disease. Alzheimer's disease typically follows a predictable pattern, with amyloid plaques forming first in the neocortex and spreading to medial brain regions, and tau tangles following the opposite pattern. Data suggests that tau tangles are more closely associated with symptomology than amyloid plaques. In early disease, tau tangles appear in the transentorhinal region, entorhinal cortex, and hippocampus, leading to disruptions in sleep, appetite, and emotional processing. During mid-stage progression, tangles develop in limbic structures such as the amygdala, thalamus, and claustrum. Amygdala atrophy is related to changes in motor behavior and potentially anxiety and irritability. At late-stage disease, tau tangles spread to isocortical areas, primarily affecting the neocortical associative areas. Severe involvement of the striatum and substantia nigra can also occur at this stage. Neocortical pathology is strongly associated with cognitive decline, and medial temporal tau tangles are associated with memory loss. Treatment of Alzheimer's disease. Use of acetylcholine esterase inhibitors or glutamate receptor antagonists to relieve cognitive and dementia symptoms are standard treatment options in Alzheimer's disease. But these treatments have not been shown to be effective in arresting disease progression. Targeting amyloid aggregation continues to be a key research direction for prevention of progression of Alzheimer's disease. Further research is also underway into other possible disease-modifying targets, such as autophagic, anti-inflammatory, neuroprotective, and anti-tau therapies. Early intervention is likely to be critical, which is only possible with a reliable disease biomarker. In addition to existing tests such as brain imaging and measurement of protein levels in cerebral spinal fluid, research is ongoing into other fluid and blood-based biomarkers and cellular changes that may be present at an early disease stage. Such research offers hope that we will be able to identify and treat disease before significant brain damage can occur.